The next section is on organizing and interpreting your results. So what happens when you start getting people saying, oh, I want to join your project? They just buy the kit, they send it off, and then 6 to 8 weeks later the results start coming in. How do you start organizing the results and grouping patients together, grouping participants together? Let's have a look at that. Just to recap on what's involved with a test, you either swab or give a sample of saliva, put it into a test tube, send it off in the post to the lab. In the lab, they look at it, they extract the DNA from the test tube, they post the results on their own personal web page. They also compare your results with everybody else's in the database and come up with a list of matches for your DNA, and they post those matches on your web page as well. And then they give you the option of joining projects that are relevant to your own particular surname, your own particular genetic signature. So that's a brief overview of the DNA testing process, and we'll be focusing on the results, the matches, and the projects today. Just to take another kind of very, very whistle-stop tour about DNA and what it's all about. When you swab your cheek cells or get a saliva sample, there is various cells in that sample. In the cell you have mitochondria, which are the source of mitochondrial DNA, and that's useful for tracing your maternal line, your mother's mother's mother. We're not talking about that today. But in the nucleus of the cell, you have 46 chromosomes arranged into 23 pairs, and the one we're interested in is pair number 23, also known as the sex chromosomes X and Y. And if you get an X and an X, then you are a lovely svelte woman. If you get an X and a Y, need I say anymore? So, and it's the Y chromosome that will actually help us trace our direct paternal line, father's father's father, all the way back to Africa about 60,000 years ago. So that's the one that we're primarily interested in. And even though the Y chromosome is relatively small compared to the others, it's still huge. It has 59 million bases, 397 genes, and 5,000 SNPs. We'll talk more about those abbreviations later on. But here is the Y chromosome. It's short arm, it's long arm. And if we unravel it, we see the classical double helix of DNA looking like, if we straighten it out, looking like a ladder or a railway track. And each of the rungs on the ladder are made up of a base pair, a pair of bases. And the bases, also called nucleotide bases, are G, C, A, T, guanine, cytosine, adenine, thymine. And G always pairs with C, A always pairs with T. You will never see G pairing with A or G pairing with T. You'll never see C pairing with A or C pairing with T. So T, A, T, C, G, T, A, etc., etc., etc. Now, just keep that in mind, because when your project member gets his results, it's important to know, from your point of view, what he's actually seeing. So another reason for doing the DNA test yourself is so that you, when you see your private pages, you will know what all your other members will see as well. So let's take a look at these private pages. And these are my private pages. And if we make this a little bit bigger on the top, we can see that under my account, I've got changed my password. The most important one here is most distant ancestors. And I find it very difficult trying to persuade people to put that in, and because this really is important. It tells you where that particular family branch is likely to come from. So that is the most important field in that particular set of pages under my account. If you look at projects, I can click on projects, I can join various projects, I can manage the projects that I have joined. If I click on resources, there's frequently asked questions, glossary, a whole load of resources here. If I click on, and this is the main area where you look for your results. Here are the Y-DNA results, these are my mitochondrial DNA results, and here's my family finder or autosomal DNA results. If we just click on Y-DNA, there's a variety of pages here, and we'll start off by looking at the actual YSTR results, the Y DNA results themselves. This is what it looks like. So let's just take a step back and see what we're looking at here. I'm going to enlarge this section here. We have various panels. Markers 1 to 12 are in panel 1. Markers 13 to 25 are in panel 2. And markers 26 to 37 are in panel 3. And we have the marker name just below that, and the value for each marker below that. Now, the way, just remember DIS 458, and I have a value for 16 on DIS 458. What does it actually mean, these markers? The way I like to visualize it, it's as if the Y chromosome is turned on its side, and all of these are the markers along its length. So what we're looking at is the Y chromosome on its side, and the various markers located along the length of the Y chromosomes represented in this row here. And then the value for each of those markers is represented immediately below that. So we've mentioned markers. Let's talk about markers, mutations, and human migrations. Two types of marker. First one is STR, the second one is SNP or SNP. STR stands for short or simple tandem repeats. It's a sequence of bases repeated many times, and it's previously thought to be junk DNA, but people are revising that quite considerably now. SNPs are single nucleotide polymorphisms, a simple replacement of a base with another base, but it can have profound effects. It's important to remember that as humans, we're around about 99.5% similar to each other, so there's only a 0.5% difference between us. But that still accounts for 30 million bases. So 0.5% is still a huge number when you actually convert it to bases. As we copy our genetic material and pass it on to our offspring, we make mistakes, and these mistakes are called mutations. And we'll look at that in a second. And they're passed on to subsequent generations. So, first type of marker, short tandem repeat, a short sequence of bases repeated many times. And here's the Y chromosome here. Here's an example of various STR markers. This is the repeat motif. And we previously mentioned DIS 458. And the repeat motif for DIS 458 is GAAA, repeated n times. And the average number of repeats is 17. Do you remember how many repeats I had?
16.
16. Let's have a look. 17 is the most common. It's 32.1%. 16, 24%. So I'm kind of in the same group as 24% of the population. It varies from a repeat of 10 to a repeat of 23. And at the extremes, very, very few people have a value for DIS 458 of 10. Only 0.003%. And very few people have a value of 23. Again, a very tiny minority of people. These would be rare marker values, 10 and 23. And that can be very useful for differentiating between different groups when we come to grouping the people together. So just keep that in mind. Single nucleotide polymorphisms, the substitution of a single base. For example, here we have a G and a C, and here it's changed to an A and a T. So the sequence here would be G, G, A, T, but down here it's changed to G, A, T. It's a very simple mutation, but it can have profound effects. It can kill people, it can stop babies from developing properly, so these SNPs can have profound effects on human development. But if we go back to early man in East Africa, and he's made the decision that he wants to copy his genetic material and pass it on to his offspring, and he's pretty successful, really. But you might notice that this child here has developed a mutation, and this one has developed a mutation. This one has a blue nose, this one is very, very tiny. But in terms of human migration, this chap might say, it's getting a bit crowded around here, I'm going to move way, way off. And his brothers and sisters say, where are you going? He says, I'm going to Norway, I don't think it'll be quite so obvious up there. And this guy says, well, I'm just going next door because my legs are too short. So they go off, and of course their offspring develop their own sets of mutations. And as more people spread out, and then people spread out, you get different mutations accumulating in the different branches. And this is what happened as man set out from Africa into Asia, Australia, Europe, and the Americas. Even though they all came from the same source, each branch, as they went their separate ways, developed and collected an array of mutations. And they've left a kind of, it's like a paper trail. And scientists are able to follow that paper trail, and together with archaeology and linguistics, they're able to trace the migration of humans out of Africa. So mutations are very, very helpful from that point of view, and there are some useful references there about that human migration and the human journey. 3 types that you'll be maybe faced with when project members contact you are haplotype, haplogroup, and haplotree. Haplotype, well, you see each marker has a variety of different versions. A haplotype just means the array of markers in a particular individual which confers a specific genetic signature on that individual. And this specific signature is called the haplotype. So we all have haplotype. It's more or less synonymous with genetic signature. So we all have that genetic signature, we all have that haplotype. Haplogroup is just a group of individuals with similar genetic signatures. And the more similar those signatures are, the more closely related people are. Each haplogroup can be further divided into subgroups or subclades, as they're called, resulting in finer and finer branches of the haplotree, which is really a diagrammatic representation of the relationship between the different haplogroups and approximates the evolution of modern humans. So it can be thought of as an evolutionary diagram of mankind, starting with genetic Adam, the ultimate family tree. And this is the sort of figure that you'll see, and it very approximates the tree with its finer branches as it goes out. The next page that I want to look at is the migration maps, the haplotree, and the haplogroup origins. And I'm going to run through this fairly quickly because Debbie will be covering SNPs later on. But this is an example of where I fit in to the human haplotree. And I'm down here in haplogroup OR. And mapping of the tree uses SNP mutations, which Debbie will talk about. So I'm just going to skip through this. But just suffice it to say that over the years, there have been more and more markers discovered, and it may be that eventually we'll just be able to have branches for individual families. So the importance is that in the next 5 years or so, SNP testing may assume greater significance for one-named studies. Just be aware of that, but we will talk about it in more depth later on. Now, and again, this is where I sit on the haplotree. These are the SNPs that have been taken. This is a more recent diagram on Wikipedia of my particular branch, and that's where I sit there. That's just for reference, really. The take-home message is, as new markers are discovered, finer and finer branches of the tree will be discovered as well.
Howard, how much convention is applied to the R1A, 1A, 2B, etc., as opposed to the L21 and so forth?
That's gone.
Yes.
So the big long string of letters is gone, and that's going to be replaced with the haplogroup, which is R, followed by the terminal SNP, the last mutation in the line. And my last mutation is Z255. So people will talk about the RL21 group, and that will indicate all of these groups down here. My specific terminal SNP is OR Z255. So I would identify myself as OR Z255, rather than OR1, B1, A2, A1, A1, B4, which is more of a mouthful. Yes?
Morris, does FTDNA give you all this information?
They do. This is all taken from the FTDNA page, other than the Wikipedia map. But you can access those graphs and charts on the Wikipedia website down there.
The SNP testing is extra. It's on top of the £80 test that you pay when you first start out.
Yeah.
I'll cover that this afternoon.
Just to say briefly, you get these nice maps. Here's genetic Adam. This is where your ancestors came out of Africa. You also get a pie chart that shows you how common the various haplogroups are in different parts of the world. You can see, for example, that haplogroup 4 is quite common in Europe, with about 45%, but it's probably absent in sub-Saharan Africa, and I don't think there's anybody over here in Siberia. So that gives you an interesting overview of the different haplogroups around the world. If you look specifically at the UK and Ireland, 4 haplogroups account for about 90 to 95% of all people, and that's OR1A, OR1B, I1 and I2B. OR1B accounts for about 3/4 of people in the British Isles. OR1A is at 4%, I1 is 8%, I2B is 4%, and others make up about 8%. And it varies between England, Scotland and Wales. You see that OR1B is 83% in Wales, 81% in Ireland, 72% in Scotland, 67% in England, and in parts of Ireland it goes up to close to 90%. OR1B can be a problem because many projects will be OR1B and nothing but OR1B, and trying to differentiate between the different subgroups of OR1B is very difficult. Debbie will touch on that this afternoon as well. So we've looked at the haplogroups, the haplotree and the migration maps, and the last thing in the private pages is the matches. And this is from my Gleason kit. And at 37 markers, I have 7 matches, but only 1 Gleason. Neville, McMahon, McLaughlin, Hart, Markham. So why do I match them? We do share a common ancestor, but it would have been before the common existence of surnames. So even though we do have a common genetic signature or a similar genetic signature, this would be way back, more than 1,000 years ago, probably. This one Gleason here holds out hope that I'm probably related to them within the last 1,000 years or so. Now, if you look up over here, there's a section that says steps, and all of these steps are 4, meaning that there are 4 steps away from an exact match. So it means that they're probably not very closely related. If it was an exact match, then it would be quite a close relationship. You've probably got a common ancestor somewhere in the last 200 to 500 years. But with a distance of 4, you're probably talking 600, 700, even more than that. So that's just important to remember, because we'll talk about distance in a while. And this is the 37 markers. Now, I've tested out to 111 markers, and some of your members will as well. But 37 markers is the recommended starting test. I've tested out to 111, and at 111, I have no matches at all. If I compare at 67 markers, then I've got 2 matches at 67. The same Gleason is appearing at 67. If I compare at 25, I've got 63. And if I compare at 12 markers, I've got 2,888. So the important thing to remember here is that 12 markers, about 5% of the FTDNA database is tested only to 12 markers, and only about 11% have tested up to 111. But quite a few people have tested at least 37 markers. If you add that up, it's around about 58% of people have tested to at least 37 markers, if not 67 or 111.
How many markers do they require to have you up for a crime?
For a crime?
But that's only 10 markers.
Is
So
that?
it is a different type of test. You use autosomal STR markers.
Oh, right.
And we only use 10.
So you
13
won't-
in America.
You won't be convicted on the basis of your YDNA test then?
I thought it might be many, many more than that.
No, it's
It's a
actually...
different kind of test.
So, as a general rule, the more markers tested, the better the estimate of the closeness of the match. But very few people have tested out to 111 markers, and only about 26% of people have tested out to 67 or greater. So, I still have to wait for people to test out to 111 markers to hopefully get a match. I'm waiting for people to join at 111 markers. So So that's the matches, and that's really a brief run through the private pages that your members will be able to view. And this is all their personal information, and only they will be able to see these private pages, and you as project admin. The last section of the private pages I want to draw your attention to is the projects that I've joined. And you know, anybody who takes a DNA test can then go on and join a project. If I click on the Spear and Surname project here, it will take me to the public pages of the Spear and Surname project website. And we've seen this before, but what I want to do now is I want to look at the Y-DNA results page. And this is the typical Y-DNA results page. In the rows, you can see genetic family 1, genetic family 2, and we have genetic family 3 here. So that the results are grouped into genetic clusters which share the similar genetic signature. There's 12 members in genetic family 1, only 1 in genetic family 2, and there's 2 members in genetic family 3. Look at this guy here. He hasn't filled in his most distant known ancestor information, so we cannot tell very much about him. He's chosen to remain private, and you'll get that kind of, from time to time, you'll find that happens. We have the most distant known ancestor in this column here. Here's my great-great-great-grandfather, Patrick Spearin, born 1802, died in 1872. I think he was born in Limerick. We've given my particular branch of the family the identification number Ireland 4 Dublin 1. But I've got identification numbers for every single one of the 236 branches in the project. Country of origin is here. Haplogroup. All of these are I2B1. These ones down here are R1B1A2. So they're still using the old nomenclature for the haplogroup on the results page. And then you have each of the markers along the Y chromosome, imagine this line on the side, and each of the values for each of the markers underneath for each of the participants in the project. So that's an overview of the results page. I tend to go, that's the classic presentation. I tend to go for the colorized one more often because that's where you see the mutations, because it colors the mutations as pink or blue blobs. So if we click on that, then here's the same kind of diagram again, but here's highlighting the mutations. All of these values here, between all of the members, are exactly the same until you get up to these markers here. And here you see that the mutations are indicated by these colors. Now, let's look at what we're looking at. This is panel 1 to 2, markers 1 to 12 rather. The first panel of markers is markers 1 to 12. The second panel of markers is markers 13 up to 25. And the third panel of markers is markers 26 up to 37. And 37 markers is your standard starting test. Now, the ones that are coloured in red are fast mutating markers. And they're much less reliable for helping you distinguish between different groups. If something's mutating very, very quickly, then they might, it may be that it doesn't help you identify who's more closely related to each other. It's better to rely on the slower mutating markers to determine who is closer to who. But you'll see here that the last member of my group has 2 mutations, the second last one has 3 mutations, and everybody else just has 1 mutation. Let's look at this second last one. It has a mutation on a fast mutating marker here. This one here, 21, whereas the average is 20, is also on a fast mutating marker. And this one here, 18, is also on a fast mutating marker. So maybe having these mutations doesn't really mean anything in particular. They would be much more relevant if there were 3 mutations on slow mutating markers, because it would take much, much longer for those mutations to occur on average. Let's look at the minimum, the maximum, and the mode. Here we have the minimum value for all of the markers, the maximum value for all of the markers, and the modal value. Now, the modal value is quite important. And mode is the most frequent value. You might be familiar with mean and median. Mean is the average value, median is the middle value in a string of numbers, but the mode is the most frequently occurring value. And the reason why the modal value is important because in most cases it probably represents the haplotype, the genetic signature of the common ancestor to all of these members here. And that's why it's sometimes called the ancestral modal haplotype. I said it probably represents because it can be skewed if there's no representation of one branch. But that's the ancestral modal haplotype, and we'll be talking about the modal haplotype here further on. So, sure.
Can I just go back
Sure.
to that? There is fourth down from the top in the right-hand section, a whole series of numbers missing. Yeah, just above the.
Oh, here, yes, that's right.
Why are there no results? Have you chosen to remove them? Or?
No, that was a transfer from DNA heritage. So somebody from a different company tested and then transferred their results over to family tree DNA. The trouble is the company that they tested with previously didn't test all the markers that family tree DNA tested for. So when they transferred the results in, some of the marker values were missing because they'd never been tested in the first place. So, we've looked at the public pages. Now let's go back to the admin pages, and specifically at genetic reports and project admin. And in the last 5 minutes of the presentation, I'll run through this. Here's the admin pages. If we click on genetic reports, I want to look specifically at genetic distance, because as the results come in for your project, they will just be en route. They won't be put into any particular group that you've created, they'll just be put into a large group called en route. And it's up to you to allocate them to a specific group. So, in this example, there are 1, 2, 3, 4, 5, 6, 7 people. The first 3 have already been allocated to genetic family 3, 4, and 1, and we've got 4 new participants who have yet to be grouped. Make sure that you set your markers to 37 and click on this arrow here. This arrow hides away all the time, and it's very, very difficult to know to click on it unless you actually have done this before. But if you click on that arrow, then what you will get is this genetic distance report. And the genetic distance has the kit number, the name of the person, the number of markers they've tested, but also the distance they are from the from this particular person here. So you can do this for every person in your project and see how far they are from every other person in your project. And this genetic distance is what we spoke about earlier on, and it's very, very important because according to the family tree DNA FAQ, frequently asked question, and there's the link down there, using the YDNA37, if the genetic distance is 0, then you're very tightly related. If it's 1, you're tightly related. If it's 2 to 3, you're related. If it's 4, you're probably related. But if it's 5, it's only possibly related. And if it's 6, it's not related at all. Now, the problem with this approach is it doesn't take into account fast mutating markers versus slow mutating markers. It just lumps them all together. And I would contend that if you have a distance of 5 and they're all in fast mutating markers, that's going to be much less relevant than if you have a distance of 5 and they're all on slow mutating markers. So, a suggested approach to this would be that you use this genetic distance guide for the first 3 categories. Use it as your first guide. But for those members who are probably or only possibly related, these ones down here, use the TIP tool, the time predictor tool, comparing it against the modal haplotype as your second guide. Now, what do I mean by the TIP tool? Let's go back to the previous slide. There's the genetic distance. This is the TIP report here. And this is the TIP report that compares this particular individual here against every other individual in the group. And if we click on the TIP report, then I'm going to click on this one here first, the one that has a genetic distance of 5. then we get this type. Well, you can also access it by clicking on genetic reports and then going to the Y-DNA tip. But if we click on the tip report, then we get this kind of report coming up. And it says, in comparing Y-DNA 37 markers, which showed 5 mismatches, the probability that Mr. Wall and Mr. Spearman shared a common ancestor within the last 4, 8, 12, 26, 20, 24 generations, 90%. So there's a 90% chance that they've got a common ancestor within the last 24 generations. 24 generations, if you allow 25 years per generation, that's 600 years, brings you back to 1350. If you allow 30 years per generation, 720 years brings you back to 1230. If you allow 33, it brings you back to 1150. That's at the level of your 22 times great-grandfather. And you've got 16 million ancestors at that particular level. So, but this is the one that you really want to look at. And if we, what I'm going to do is I'm going to look at the tip report for each of these in turn. We've got a distance of 5, 5, 6, 7, and 12. We've loaded up a bit, 5, 5, 6, 7, and 12. And the tip report at 24 generations comes back at 9%, 9%, 75%, 60.7%, 9.6%. So the tip tool is just another way of looking how close or how distant people are related to each other. The threshold that we use is 60%. So at 24 generations. The threshold for inclusion in the same genetic family is 60%. This is what was developed by James Irwin in his project, and I'll show you the reason for why in a second. But it means that there's a 60% or greater probability that the individual shares a common ancestor with members bearing a modal haplotype within the past 24 generations. Now, supposing there are still borderline cases, and that 60.7% is bothering me because it's so borderline. What can you do then? Well, you consider using the tip tool at 67 or 111 markers. But not everybody will have tested out that far. You can assess the people involved for rare marker values. If they both share a rare marker value, then the likelihood is that they probably are in the same genetic family. But the real clincher, the real decider would be to do formal SNP testing, and that would be extra on top of what you've already done. So here's what we did previously at 37 markers. We've got 90, 90, 75, 60, and 9.6. At 67 markers, we can see that even though the genetic distance was 5 at 37, and it got very, very similar results with the tip tool, those results have really gone down when you go up to 67 markers. And it just goes to emphasize that the more markers you test, the more accurate the estimate of the closeness of the relationship. We can't do it for these two because they haven't tested to 67, they've only tested for 37. But on the basis of the tip 67 analysis, only this person would be included in the same genetic family. These two people would be excluded from the same genetic family. So that's the tip tool. And this is a graph from James' project, and it shows the justification of the 60% cutoff. There's 60%, this goes up to 90%, and down to 0%. But the 60% will give you the greatest demarcation between including people in a genetic family and excluding people from a genetic family. It really helps to differentiate and make a clear distinction between two groups of people, those who belong and those who do not belong. And you can see that even then there are still some possible borderline cases here, and SNP testing or rare marker values might be the way forward with these particular individuals here, but this is going to work for most people. So, to recap then, and this is my last slide, just to show you how to do member subgrouping, I'll run through it very, very quickly. You know which groups they should belong to. How do you actually do it? Click on member subgrouping, click on create subgroup, and you can name your subgroup, provide a description for it if you want to, and you can even color code it. And I've created genetic family 1, 2, 3, 4. I've provided a little description there about different colors for them. But in order to manage them and to allocate people to different groups, you again have to click on this tiny little arrow, this deceptive little arrow. And by clicking on that, it takes you to the Add or Remove page. And I click on the arrow for genetic family 1. You can see there's 3 people already in genetic family 1. I can remove any of them if I've changed my mind and they shouldn't be there. And these new ungrouped people, I can decide whether or not they need to go into genetic family 1 on the basis of my analysis of genetic distance and using the tip tool. So that really is member grouping complete. And by doing that, you will be able to set up and run your project at a very competent level. The only other pages I want to draw your attention to are the member information pages. That's how you access the private pages of all the members in your group. In terms of genetic reports, we've already looked at genetic distance and tip. Those are the most important pages there. In the project administration pages, the member subgrouping, what I've just shown you, is important, and we've already set up the project profile and the public website. And now I think it's time for coffee.